In this video, we're going to talk about the cell cycle control systems. So what I mean by this is, let's say you have a cell, and you can just envision it being a skin cell, just so you can visualize it as something. And now normally your skin is replenishing itself at a consistent rate. So every 12 hours or so, your skin cell will divide, and then another 12 hours goes by and these guys divide. What would happen if your skin cell suddenly started replicating out of control, and maybe instead of every 12 hours, it was replicating every 4 hours, right? You would have this uncontrolled skin production, and cells would just start piling on top of each other because cells are not dying fast enough and these new cells are being made and they're piling up. So that could lead to something called a tumor, which is bad. You don't want that. And this would be basically uncontrolled cell growth if cells are growing too fast. Or you could have the opposite effect where maybe a skin cell is dividing too slow. Instead of dividing every 12 hours, the cells are dividing every 48 hours. That would mean your skin's not healing itself. It's not replicating enough. That would be a problem too. So the cell cycle needs to be very controlled. It needs to replicate cells using the proper time. It needs to not replicate them too fast or too slow. But even more important is that the cell needs to make sure there's no major mutations in the skin cell. Like, what if this parent skin cell had an extra chromosome? Instead of having 46 chromosomes that had 47, or even less chromosomes that had 45, and then it started replicating, and then these cells would become mutated. The cell needs to really make sure everything is good, everything's looking good, like a checkpoint to make sure that there's no problems. Otherwise, this is how cancer is formed, right? And this skin cell example I gave, it's about 12 hours for a skin cell to divide, but every cell is different. So for example, your neurons, they never divide. Embryonic cells may divide every 2 hours instead of every 12 hours. Red blood cells may divide every 20 hours. So every cell in the body is different. This was just an example. So in the cell cycle, there's 3 checkpoints to kind of make sure everything's going okay. So I always like to start the cell cycle. I mean, it's a cycle, so there's really no starting point, but in my brain, it starts here at G1. This is when you have a fresh, brand new cell, and during G1, it's going to start thinking about dividing. It's going to start preparing some enzymes potentially for DNA replication during the S phase, and it's going to start replicating some cell components like mitochondria and ribosomes and things like that. And although all the checkpoints are important, I think the G1 checkpoint, in my opinion, is the most important, because this is where it all begins. If a mutated, damaged cell accidentally makes it past G1, it's almost like going to be really hard for it to not replicate at that point. So this is, in my opinion, the most important checkpoint. So during the G1 checkpoint, the cell needs to make sure that there's no major DNA damage. It needs to make sure that enzymes are prepared for DNA synthesis, and it needs to make sure there's enough cellular components and organelles in the cell to proceed with cell division. If everything looks good, we get the green light and the cells can continue on into S phase. If there's a huge problem, something does not look right, then the cell can enter something called G0. And this only occurs during G1. So it's either go after G1 or go into G0. In G0, the first thing the cell's going to do is try to repair. It's kind of like if you had a car and it's not working, it's broken down, you're going to bring it to the car shop to try to repair it. So that's your G0, the car shop. So first, the cell's going to try to repair itself. If it can repair, it can leave G0, go back into G1, and proceed onto S. But sometimes it doesn't get repaired. Like, haven't you guys had that car that just kept having problems and you kept putting money into it and nothing you could do would repair that car? So what did you have to do with that car? You had to get rid of it. So the same thing with the cell. If the cell can't repair, then it might do something called apoptosis, which is basically a programmed cell death. And basically what that means is the cell will destroy itself so that it doesn't replicate. sometimes a cell is in G0 for so long, it's just trying to repair itself so long that it kind of gets stuck there forever. It doesn't undergo apoptosis, but it never actually gets repaired. It kind of just gets stuck in this permanent G0. So for example, that's what happens with the neurons. They enter this G0 state and they never come out of it. They're just stuck there forever. So that's the first checkpoint, the G1 checkpoint. Now, if the cell passes the G1 checkpoint, it's going to go into S phase. Remember, during S phase, it's going to replicate the chromosomes, replicate all the chromosomes. And then it's going to proceed to G2. And remember, in G2, this is kind of the place where the cell is going to prepare for mitosis. Enzymes are going to be made to begin mitosis. Now, at the end of G2, before mitosis starts, that's where our second checkpoint is. So the second checkpoint is the G2 checkpoint. Now, the G2 checkpoint is checking to make sure the DNA replicated properly. So what that means is, let's say you have the cell, it's getting ready to go to mitosis, and you have 46 chromosomes, but during the S phase, all the chromosomes got replicated. So now there's a copy of all the chromosomes. But let's say one of the chromosomes didn't replicate itself, or multiple chromosomes didn't replicate itself. This would be a problem because this would mean that the new offspring wouldn't have the proper number of chromosomes. So that's what G2 is checking for. It's checking to make sure that the DNA all replicated properly. Again, if it did replicate properly, everything looks good, we're going to go on to the next phase. But if it didn't replicate properly, then the cell is going to have to try again to repair itself. And if it can't repair, it's just too damaged, then apoptosis. The final checkpoint is in mitosis. It's the mitotic checkpoint, or the M checkpoint. This checkpoint happens between metaphase and anaphase. So remember, in metaphase, all the chromosomes are going to line up in the center of the cell. Then you have these spindle, the microtubules, that are going to connect them. And remember, then in anaphase, the next step, the chromosomes are going to get pulled apart. So what's happening in the M checkpoint is they want to make sure the chromosomes lined up perfectly in the center. If some of the chromosomes didn't line up, right? They didn't line up properly, or they weren't attached at the spindle, that would be a problem because that would mean the new cells wouldn't get the proper number of chromosomes. So once again, M checkpoint is checking to make sure that the new daughter cells will have the appropriate amount of DNA. If there's something wrong, they need to try to repair it. If they can't repair it, the cell will undergo apoptosis. So you can see these checkpoints are so important for ensuring that the somatic cells don't have these major mutations that will spread to the new daughter cells. How does a cell know whether to go or to stop? Like, how does it know whether there's a huge mutation or whether everything is good? So there's a couple of proteins. It's always proteins because they do everything, right? There's a couple of really important proteins and we need to know about them. So the first, let's talk about the go signals. The go signals are known as CDKs and cyclins. And then we're going to talk about the stop signals. What will stop a mutated cell from replicating and forming a tumor? Well, that's a group known as the tumor suppressor genes. Just like it sounds, these genes are going to try to prevent tumors from forming. And we're going to talk about a couple of them. We're going to talk about P53 and RB. So the go signals of the cell cycle, right? What keeps the cycle going throughout are mainly these things called CDKs. CDK stands for cyclin-dependent kinase. Hey, I think you guys learned about kinase before. Remember, a kinase is simply a protein that can add phosphates. They can stick phosphates onto other proteins and either activate the proteins or deactivate them. So during the cell cycle, you'll have this CDK molecule. They made it look like a big Pac-Man here. So when the CDK is all by itself and lonely, it's in the inactive form. So when it's alone, it's inactive, turned off. The way to turn a CDK on is it needs to bind with its friend. Now it's not lonely anymore, okay? And its friend that's going to bind and activate the CDK is known as a cyclin. So a cyclin is a protein, and just like it sounds, there's many of them throughout the cycle. Cyclin indicates cycle, so there's multiple cyclins throughout the cell cycle. And so during the cycle, the cyclin will bind to the CDK, and when they bind, this will activate the CDK. So in order for the CDK to be active, a cyclin will bind to it. So let's walk through the cycle, and we'll just keep it this simple, okay? So remember, I like to start here at G1. So here's the CDK. Right now it's turned off because you can see it doesn't have any friend, it's all alone. Now we're going to enter G1. During G1, there's something called the G1S cyclin. This is like basically the start cyclin. So the cyclin will bind to the CDK. They'll bind together. Now the CDK is happy and it's turned on. If the cell makes it through the G1 checkpoint and it's going to proceed to the S phase, what will happen is this first cyclin, the G1S cyclin, the pink one, will fall off the CDK and it'll get all chopped up. So we're done with that first cyclin because we're done with G1 phase, so that first cyclin will get chopped up. And then once again, the CDK is all alone because it lost its friend. But now another cyclin comes in. This time it's the S cyclin. So the S cyclin comes in, binds CDK. CDK is happy again. It can do its job. It's active. However, at the end of S, now that S cyclin is going to get chopped up and degraded because we don't need it anymore. We're going on to the next phase, G2. Now in G2, there's another cyclin called G2M cyclin. This teal one, that one's going to bind. That one will stay bound throughout mitosis, at which point that cyclin will come off, get degraded, and we'll end up with a lonely CDK again, and we'll start the cycle over. The overall takeaway from this, CDKs are inactive when they're all alone. They become active when cyclins bind to them. There's different cyclins throughout different phases of the cell cycle, but at the end of each phase, that cyclin will fall off, get chopped up and degraded, and the new cyclin can come in and do its job. Now, like I said before, I think the most important step in the cell cycle is this first step right here, the initiation of the first cyclin, the G1S cyclin, binding to the CDK, because that's really what gets the cell cycle going. So let's kind of zoom in on this first step. Take a look at it. So this is zooming in on the signals that initiate the cell cycle and tell it to go, tell it to turn on and to divide the cells. So here you have a cell, could be a skin cell, whatever. Right now it's not dividing. It doesn't really know if it should divide or not. It needs to be told, hey, you need to divide. So in the environment here, there's these different molecules. These are called mitogens. So in order for the cell to divide, the first thing that happens is one of these mitogens need to come in and bind to a receptor on the cell. So over here you can see, here's the cell, here's a receptor. The first thing that happens is a mitogen binds the receptor. This sends a signal into the cell and it'll initiate the first cyclin here, the first one in the G1 cyclin, to bind to the CDK. So I'll write it down. Step 1, mitogen binds the cell receptor on the outside of the cell. This will send a signal allowing the G1 cyclin to bind to the G1 CDK. Now remember, when the cyclin binds the CDK, this CDK is now activated. Once the CDK is activated, many things are going to happen, but one of the most important ones is this step. The CDK is a kinase, so it can phosphorylate things. It can add phosphate groups to things. Inside the cell, there's this protein called the RB protein. It's this big red one. The RB protein is a tumor suppressor. When the RB protein is active, it'll block transcription of cell division genes. So in the normal active state, the RB is this like big anchor. It's blocking transcriptions of genes needed to replicate the cell. When this first cyclin and CDK become active, this CDK is going to tack on phosphates to the RB protein. When this happens, RB gets released. It pops off. So as you can see here, the RB protein gets phosphorylated and it pops off. When the RB protein is phosphorylated, it's now considered inactive. Once this RB protein pops off, now transcription is free to happen of these S-phase genes so that the cell can replicate its DNA. So in step 3, the CDK phosphorylates the RB protein. The RB protein pops off and transcription of these S-phase genes can occur. So let me walk through one more time. Mitogen binds the cell, signals come in, cyclin and CDK bind together. This activates the CDK. CDK adds phosphates onto the RB protein. The RB protein can pop off. This frees up RNA polymerase to come in and transcribe genes necessary for cell division. Now the RB protein is considered a tumor suppressor because it's preventing transcription of cell replication genes. So meaning if you have a cell, if the RB protein is active, it cannot divide at all. The RB protein's blocking cell replication. But if the RB protein is inactive, now the cell is free to transcribe those genes and the cell can divide. So in the active state, cell can't divide. If the RB is inactive, it can divide. Let's say there was a mutation in the RB protein so that the RB protein could never bind here. It was unable to bind and the RB protein was constantly inactivated. If the RB protein was constantly inactivated, and I'll put a little star here, whenever you see that star, it means mutated. So if that RB protein was mutated and was constantly inactivated, that means the cell could constantly undergo transcription of these cell replication genes. And the cell could just start growing out of control. You'd get a tumor. In fact, there are cancers out there where an RB protein has been mutated. Transcription of S-phase genes is unlimited and cells divide out of control, forming a tumor. Luckily, the cell kind of has a backup mechanism. In case something went wrong and the RB protein became inactivated when it shouldn't be, there is a backup or a stop mechanism, okay? So we need to stop this out of control cell replication before it becomes a tumor. And this stop is another tumor suppressor gene. And this one is called P53. So here's how it works. Let's say that you have really damaged DNA, okay, a big mutation. For example, if someone gets an X-ray, it can cause a double-stranded break in the DNA. That's bad damage, right? So whenever there's damaged DNA, this will lead to activation of P53. The way that P53 becomes activated is it becomes phosphorylated. So if there's no phosphate, P53 is inactive. If there is phosphate, P53 is active. So activation of P53 means P53 becomes phosphorylated. And the only way P53 becomes activated is if there's damaged DNA. Now what will happen is the P53 will bind to the regulatory region of a gene called P21. So basically P53 is an activator. Remember when we talked about activators last week? P53 is an activator of the P21 gene. So P53 will turn on transcription of P21. You'll make P21 mRNA, and then that P21 will be translated into the P21 protein. The P21 protein, it inhibits CDK. So normally, if CDK were active, it can go around phosphorylating things. But P21 clamps CDK so that it is inactive and it blocks it from doing anything. Okay, so it's kind of blocking it here. So let's put it all together. If there was something wrong with the DNA, it was damaged. We don't want this CDK activating RB, which allows RB to be pulled off and for transcription to happen. We don't want transcription to happen. So what we could do is P53 could activate P21. P21 could clamp this CDK, preventing it from working, block it. So P21 will block CDK. CDK then can't phosphorylate RB. Then RB can never pop off and we don't ever get transcription because we don't want to replicate the cell if there's major DNA damage. So this is basically how we can turn off cell division. We can block the cell from passing G1 phase and the cell can then go into G0 and try to repair any damage. If the cell cannot repair its damage, like I mentioned, it'll undergo something called apoptosis, which is a programmed cell death. So during apoptosis, the cell will basically turn into these blebs, and then these blebs can be eaten up by phagocytes. Phagocytes are these types of cells in the body that could come in and degrade any junk that's left. So now we talked about how the cell controls its cell division, turns it on or turns it off. And in the next video, we'll talk about what happens when the cell does not control its cell division and when things go wrong.